Nick Majerison here. Welcome to Top Med Talk. Thanks for downloading the podcast. If you're enjoying what you hear, do make sure you've subscribed. This week we're presenting to you our Xtreme Everest mini-series, which we streamed on the website live from Duke University in America on June the 14th and the 15th when these presentations first took place. Today's piece is presented by Andrew Murray. It's called Metabolic Adaptation to Hypoxia. Have a listen.
Top Med Talk.
Thank you very much. I'm aware I'm standing between you and afternoon tea, and talking about metabolism is perhaps not the most riveting subject, but hopefully you've got a picture from the previous speakers that it is an important part of the story for us. You've seen this slide a number of times already, and I only put it up to illustrate that the classic story, the classic picture of acclimatization isn't incorrect, but it doesn't fully explain the metabolic changes that are happening at altitude. And this area has been somewhat neglected to this point, although it's beginning to receive more and more interest. And now I would say there is a real acceptance that the acclimatization doesn't just involve changes in convective oxygen delivery, but changes in oxygen utilization at the tissues. And our first indication that something was going on was from our 2007 expedition. I was a postdoc working in Oxford at the time, and we were using NMR imaging and spectroscopy to get a handle on cardiac function and cardiac energetics. So function we can understand through NMR imaging, but using spectroscopy, particularly 31-phosphorus spectroscopy, we can get an idea of what's happening to energetics in the heart, in skeletal muscle and other tissues. And in the heart, we use an index, the phosphocreatine to ATP ratio. The spectroscopy technique we were using doesn't give absolute values of these intermediates, but phosphocreatine being the energy buffer that buffers ATP levels in the heart and in skeletal muscle means that we can express these two intermediates as an index of phosphocreatine over ATP, and it gives us a decent index of cardiac energetics. My former colleague at Oxford, Stefan Neubauer, and others have really pioneered the use of this technique to understand the metabolic perturbations that happen in the hearts of patients with heart failure, diabetes, and obesity, and so on. What Stefan described is that a fall in this phosphocreatine to ATP ratio was the single best predictor of mortality in patients with dilated cardiomyopathy, for instance. So here we used it to look at, and this is data not from the individuals who summited Everest in 2007, but a group who trekked to Everest Base Camp, and we found that upon return to sea level, so these are the pre-trek values on the left, the post-trek values in the middle, we found that in every single individual, the cardiac phosphocreatine to ATP ratio had fallen upon return to sea level. And it's worth bearing in mind that these are not hypoxic individuals any longer. We didn't take the magnet with us to Everest Base Camp. Even the most sort of compliant yak isn't going to lug this up to base camp for us. So this is upon return to sea level. They're breathing sea level air. They're acclimatized to altitude. They've got elevated hematocrits, oxygen contents which are much higher than they were before they left, and yet phosphocreatine to ATP was low. The good news is, and good news because I was one of these subjects, is that 6 months later, when we rescanned everybody, this had returned to normal. So it's suggesting that there is metabolic reorganization going on in the heart. Just to show you really that this isn't just an abstract measurement, we also measured cardiac function using echocardiography and MRI. We didn't see significant differences in systolic function in this group, but in the diastolic function, which of course is not a passive relaxation of the heart, but a hugely energy demanding process, ventricular filling, left ventricular filling, was impaired upon return to sea level in the yellow bars, but at the 6-month follow-up mark, alongside the energetics, had returned to normal. So my main interest is in mitochondrial adaptation in tissues in response to health and disease. Mitochondria are, of course, interesting because, as Mike has pointed out in the earlier slide, this is where the vast majority of oxygen in our bodies is consumed at a single enzyme, complex 4, sitting in the middle of the image here, complex 4 at the respiratory chain. So it stands to reason that if oxygen utilization is being altered, then the mitochondria are a sensible place to look. And if we're thinking about the metabolic or the molecular responses to altitude, then we need to understand the transcription factors and molecular pathways that are governing these changes. And we turn our attention to the hypoxia-inducible factors. And these transcription factors are continually expressed in most of the tissues of our bodies. So in almost all of our cells right now, we're expressing HIF1 alpha and HIF2 alpha. These proteins are being produced, and yet they're being degraded, and they're being degraded in an oxygen-dependent way. So in conditions where PO2 is relatively high, HIF1 alpha and HIF2 alpha are hydroxylated, they're degraded, they're silent, they do not affect gene transcription. And this seems wasteful, except, of course, that when hypoxia occurs, this degradation no longer occurs, they're immediately active, and they can alter the transcription of genes that include some of those that we've already heard about, erythropoietin, which drives the increase in red cell production, so erythropoietin at the kidney, VEGF, which drives the production of new blood vessels, particularly the microcirculatory vessels that Dan mentioned, but also a huge number of metabolic mediators, metabolic enzymes. So we have glucose transporters and glycolytic enzymes, which all hint towards increased glucose metabolism. So a large amount of work was done on the hypoxia-inducible factors by Greg Semenza and others in the 1990s, and showed that these in cells in culture, the hypoxia-inducible factors play an enormous role in governing the metabolic response to hypoxia. So as you've seen in normoxia, they're degraded, they're inactive, but in hypoxia, on the right of the image here, a number of pathways are upregulated, driving your metabolism away from an oxidative phenotype where the mitochondria are playing a key role towards a glycolytic metabolic phenotype, which is not dependent upon oxygen. So we see a large number of the glycolytic enzymes upregulated, so the glycolytic pathway is more active. Moreover, the entry of pyruvate, an end product of glycolysis, into the mitochondrion is inhibited via the phosphorylation of pyruvate dehydrogenase kinase. So the substrates themselves are diverted away from the mitochondria. And there were further suggestions, not just in cells, but in some animal models, that factors that govern the degradation of mitochondria, mitochondrial autophagy, were also upregulated. So I've illustrated BNIP3 here, which seems to come up time and again when we look at these regulators. So with enough time in hypoxia, you might actually start to degrade and break down your mitochondria. Now, this is all very well and good for cells or for animal models, but what is happening in vivo in humans? We did start by looking at animal models, and a technique we use in my lab very frequently is to probe mitochondrial function using measurements of oxygen consumption, and I'll talk through it briefly, hopefully clearly here for you. We can look at a number of tissues using this technique, but skeletal muscle and heart are particularly tractable, and we will take our heart or skeletal muscle biopsies. We treat them with a detergent such as saponin or digitonin. These are plant-based detergents that form pores in cell membranes in conjunction with the cholesterol that's already present in that membrane. Now, this is particularly useful for mitochondrial biologists because cholesterol is present at about 1,000-fold higher concentrations in the cell membrane compared with the mitochondrial membrane. So if we get the concentration of our detergent right, we can selectively permeabilize the cell membrane, but leave all of our mitochondria intact. And we can actually check that in our assay. And so once we've permeabilized our cell, we can wash out all the cytosol, wash out the metabolites and intermediates that are already present in the cell, and then reintroduce in a stepwise fashion our own substrates and inhibitors to probe different aspects of mitochondrial function. And it's an extremely neat technique. All you're measuring is oxygen consumption, but you effectively measure it under different conditions, and it can tell you a number of things about substrate preference, capacity of the mitochondria to consume oxygen and substrate, but also the efficiency of the mitochondria. So a typical assay might involve us probing, for example, the different electron entry points, the electron transport chain. We can give substrates such as pyruvate or glutamate that normally feed electrons to complex 1, succinate that feeds electrons to complex 2, or we can probe the electron transferring flavor protein, which receives its electrons from fatty acids. So we can start to differentially scrutinize fat metabolism compared to, say, pyruvate metabolism. And we can also look at any problems that are happening in the electron transport chain. Moreover, we can also measure this under a number of respiratory states. So we'll initially start off measuring oxygen consumption in the absence of ATP production. So if we don't add any ADP, the diphosphate, it means that we don't have activation of the ATP synthase. We get a low rate of oxygen consumption. And this is proportional just to protons leaking back into the mitochondria. So, if you will, this is an inefficient use of oxygen. It's the oxygen being consumed by the mitochondria for purposes other than ATP production. So next we'll stimulate the mitochondria with ADP. This activates oxidative phosphorylation. We get a much higher rate of oxygen consumption, and this is commensurate with the rate of oxygen consumption, or the maximum rate of oxygen consumption for ATP production. So this is our efficient use of oxygen, if you will. And just for completion, we can uncouple the mitochondria and get a saturated rate of consumption, but I'm not going to say a great deal about that today. So we probed this in my lab initially in the hearts of experimental animals. Apologies to Mike, who's not a fan of rat bothering, but this was very informative for us. We went through one of these typical titrations. We get a range of oxygen consumptions here, but the ones I just want to highlight, the second group of bars in here, the white bars are normoxic animals. The paler blue, 2 days at 10% oxygen, so perhaps 2 days at Everest Base Camp, and then 2 weeks, 14 days at 10%. And the second group of bars in here is the maximum rate of fatty acid oxidation in the hearts of these rats. And you can see no change after 2 days, but after 2 weeks, this is beginning to fall. And the other major hit we see is a hit on complex 1 of the electron transport chain, although when we isolate complex 2, that looks relatively normal. So 2 clear targets here, fat oxidation and complex 1 function. So I'm going to introduce you now to my favorite metabolic regulator. This is called PPAR alpha. It's a transcription factor in the heart, in skeletal muscle, that is a master regulator of fat metabolism. And it was an obvious place to look when we're seeing fat metabolism being impaired in hypoxia. And the way PPAR alpha normally works is that it's activated by fatty acids. So the more fatty acids you have present, and this might be elevated if you eat a high-fat diet or you're diabetic or obese, the more PPAR alpha is activated and the more it turns on genes controlling fat metabolism. So it makes logical sense from a supply and demand perspective. You have more fats present, you increase fat oxidation. What we saw in the rat heart, of course, was decreased fat oxidation in hypoxia. And when we came to scrutinize PPAR alpha and a number of its downstream targets, we found that expression of PPAR alpha itself on the left of this bottom image was not affected, but the downstream targets were decreased after 2 weeks. So hypoxia is decreasing the activity of this key transcription factor. So moving on, what happens then in humans at altitude? Well, in 2007, this was our lab at Base Camp, and here's the team. Here's Dan, fresh back from the summit of Everest, looking absolutely delighted to be giving a muscle biopsy with Chris, taking this from his leg here, from his upper thigh. We studied 2 different groups. The first group was the group that trekked to Everest Base Camp. This was early in the expedition. They'd arrived at Base Camp and went no higher, and there weren't stark differences in these groups. We're looking at muscle fibers here in cross-section, with the label M indicating the mitochondria in the muscle. And when we quantified this, we didn't actually see any changes in mitochondrial density. So arrival at Base Camp, maybe 2 days, no great difference, although a trend downwards in our mitochondrial density. We did, however, see much more dramatic differences in the group that ascended to the summit of Everest, returned to Base Camp, and gave their second biopsy upon returning from Everest. And here we saw a stark loss of mitochondria in the skeletal muscle. These are beautiful electron micrographs generated by our collaborator Hans Hoppler in Bern. And you can see, I mean, it's like a very expensive game to spot the difference. You can see an area of damage here on the right. This is a poorly defined substance called lipofuscin, which we see accumulating in muscle with oxidative stress. But you can see that the mitochondria in the sample in London are much more prevalent than they are in the sample at Everest Base Camp after returning from the summit here. Those mitochondria that remain look rather smaller and more fragmented. It's a common stress response in mitochondria to fragment. This is a network we're dealing with, not individual organelles. And the network does fragment a little. But what's really striking to me is not necessarily the loss of mitochondria per se, but the fact that it's not uniform right across the cell. When we look at skeletal muscle, we find 2 subpopulations of mitochondria, a population that lies along the cell membrane, the so-called subsarcolemmal mitochondria, and a deeper lying population, the interfibrillar mitochondria. Now, it's not entirely clear and very difficult to work out functionally what the purpose of these 2 subpopulations are, but what we see at altitude is a stark loss of that subsarcolemmal population and a relative preservation, although still a loss, of that deeper lying population of mitochondria. But this almost seems not to make sense. You're losing mitochondria from next to the capillary, next to the oxygen source, but preserving them deeper within the core. That may not seem to be very sensible because the oxygen tension is slightly higher here, but you do need mitochondria deeper in because it's not just about getting oxygen to the mitochondria, but distributing the products of the oxidative phosphorylation, the ATP and phosphocreatine. But what's really striking to me is that this seems to be regulated. And when we quantify this, we see that the loss of the subsarcolemmal population is at around 73%, whereas it's about 14% of the deeper lying mitochondria, 21% overall. Moreover, it's not simply the case that you've got fewer mitochondria. When I started to look at some of the levels of various proteins and regulators within those that remained, again, we see some differences. So if we looked at the electron transport chain, we saw a stronger loss of two complexes, in particular complex 1 and complex 4, but yet again, complex 1 coming out in our study. And this is interesting because it's one of those entry points for electrons into the chain, but also a source of those reactive oxygen species that Mike alluded to in his talk. So perhaps this is a protection against that free radical production. And when we measured fatty acid oxidation, this fatty acid oxidation enzyme, we saw a very strong loss of fatty acid oxidation enzyme capacity, much greater than the loss of mitochondria. So it seems to be not just fewer mitochondria, but a particular downregulation of fat metabolism. Now, there have been previous studies. This is a much older study from Hans Hoppler, Paolo Cerretelli, and others that looked at mitochondrial density in muscle post an expedition. This is over 6 weeks at over 5,000 meters, with some subjects ascending above 8,000 meters. And here, looking at the volume of mitochondria per muscle fiber, they're seeing, again, about a 20% loss, so very similar to our data. What's slightly more puzzling, and Dan's mentioned the Operation Everest 2 chamber simulated ascent of Everest in his talk, is that in fact they saw no difference in mitochondrial density per fiber volume. And this seems to make very little sense. Not only does hypoxia affect mitochondrial function, but training status and exercise increases mitochondrial density. And you would think that people would be more active on a mountain than they would in a chamber simulated ascent. So this is very difficult to explain, but I put it there for completion. So perhaps there are differences between chamber studies and field studies. Now, it's not just about density, of course, and these are some studies that were carried out by Carsten Lumby and others looking at using the same techniques as we've used, measuring oxygen consumption, to get a handle on what's happening to mitochondrial function at altitude. And here we see that there was no difference in this particular study. This was carried out at 4,500 meters with the subject spending 9 to 11 days. Paradoxically, this is at a lower altitude of just 3,400 meters, where there was a loss in mitochondrial capacity in many of the measurements, again, including complex 1. However, this was carried out over a longer period of time, 28 days. So it's not just absolute altitude reached, but the length of time one spends at altitude. And more we're seeing that these are time-dependent changes as much as hypoxia-dependent changes. So in 2013, we returned with the team, and with the aim of studying the Sherpas, we're a bit more sort of geared up to study mitochondrial function in the field here. We aim to actually probe not just density this time, but mitochondrial function using this respirometry technique. And when we look at the Sherpas, we see adaptations all the way from source to sink, many of which you've already heard about. But strikingly for us is that there were hints that mitochondrial density in Sherpa muscle was lower than we find in lowlanders. And you've already seen a version of this slide from Mike. Sherpas on the right here compared with Messner, Hapler, and other elite lowland mountaineers and sedentary lowlanders. Sherpas' VO2 max is not remarkably high at sea level compared with these individuals. Capillary density perhaps slightly on the high side, but mitochondrial density somewhat towards the lower range of these individuals. So a hint that Sherpas actually have a lower mitochondrial density. Very interesting for us was a number of studies that came out around the time we were planning the 2013 expedition, looking at the genetics of Sherpas and Tibetans and really trying to pin some of these interesting phenotypes onto genetic variants. And the one genetic variant that comes out very reliably in most of the studies of Sherpas and other Tibetan populations is a polymorphism in the gene encoding HIF2 alpha, so the EPAS1 gene. This is very strongly associated with the Sherpas having a lower hemoglobin concentration at any given altitude than lowlanders. This came out in a number of studies, but one gene that didn't crop up in all studies but was of real interest to us was the gene encoding our favorite transcriptional regulator, PPAR alpha, this controller of fat oxidation. And there were genetic variants in the Sherpas, polymorphisms in the PPAR alpha gene that were more concentrated. So we set out to study our Sherpas and our lowlanders on the ascent, the classic ascent profile. Lowlanders gave biopsies in London prior to departure, the Sherpas in Kathmandu, their home altitude. And these Sherpas were not highland-dwelling Sherpas. They're living at relatively low altitudes and hadn't been to high altitude for at least 9 months prior to the expeditions. They were not acclimatized, although they would have the benefit of many thousands of years residence as a population at altitude. Both groups followed a very similar ascent to base camp and gave a second biopsy upon arrival, so the lowlanders and the Sherpas, who followed through on 3 separate treks. The Sherpas then went home. The lowlanders remained, these particular subjects remained at base camp for the 2 months of the expedition. We had the opportunity to study them at a later time point, 2 months down the line, to see if this longer-term acclimatization was associated with any further difference in metabolic functions. The lowlanders gave a second biopsy. First thing we looked at upon return was the genetics of these two groups, and here these pies are indicating 3 different single-point regions on the PPAR alpha gene where there are known polymorphisms, the polymorphisms described in the previous Simonson study. Within the pie, a black region indicates the individual who is homozygous for the supposedly advantageous allele at altitude, and you can see that certainly on all 3 points, the Sherpas have a great sort of concentration of individuals who are homozygous for this allele. The grey areas are those who are heterozygous, and the white areas are those who are homozygous for the supposedly non-advantageous allele. So it's not that these polymorphisms are not present in lowlanders. There are a number of heterozygotes, but those who are homozygous are very much more common in our Sherpa group. So we took the biopsies, as mentioned in London, Kathmandu Base Camp, permeabilized the fibers as described, and used the Ouroboros oxygraph as a way of measuring oxygen consumption. And we were very kindly loaned 4 of these oxygraphs by Ouroboros instruments, and they worked extremely reliably all the way up to base camp. Our students also worked extremely reliably. These are 2 of my PhD students at the time, James and Alex, using the oxygraphs base camp. And I think on this particular day, they're measuring their own muscle biopsies. So there's real dedication to the study, not just to go along and have a trip, but to literally give of yourself for the study. So very remarkable work. To accompany this respiratory data, we also measured gene expression, enzyme activities. We used high-throughput metabolomics in collaboration with Jules Griffin's group in the biochemistry department back in Cambridge. And we carried out glucose tolerance tests in the field to really get a full picture of what is happening with substrate metabolism. This first slide is showing hypoxia markers. You can see the lowlanders in blue, the Sherpas in red. As the group ascended to base camp, both groups' arterial oxygen saturations fell. So they were at a similar point at the point at which the biopsy was taken at base camp. On the right here, in the next few graphs, we'll all take this format. We have the lowlanders on the left, the Sherpas on the right. The clear bar is the lowlanders at baseline, the light blue upon arrival at Everest Base Camp, and the dark blue after 2 months at Everest Base Camp. The Sherpas are the hatch bars. This white hatch bar is the Sherpas in Kathmandu, and then the Sherpas upon arrival at base camp. And what we have here is the expression of VEGF. This is a gene that's upregulated by HIF1 alpha in hypoxic conditions. And what it shows is that the Sherpas do respond to hypoxia. It's not a case that the Sherpas don't respond. In both the lowlanders and the Sherpas, there was an upregulation of this gene upon arrival, so clear response to hypoxia in the muscle. We looked at PPAR alpha and a number of fatty acid genes. In our lowlanders, there was no change in PPAR alpha expression with time at altitude. Neither was there a change in the Sherpas, but the Sherpas started from a lower baseline with about 50% of the expression of PPAR alpha. So we can see that this polymorphism, which is more common in the Sherpas, is associated with lower expression of the gene. This is a downstream target of PPAR alpha. CPT1 is a fat transporter, transports fat into the mitochondria. We find that it's lower in the Sherpas in conjunction with the lower expression. In the lowlanders, despite the fact there's no change in PPAR alpha expression, CPT1 comes down. And this is very, very similar to our rats in hypoxia chambers, where there was no change in PPAR alpha expression, but decreased transcriptional activity. So this is 2 different ways of getting to the same route. The Sherpas are adapted. They have lower expression of PPAR alpha and lower fat oxidation enzyme capacities prior to departure, whereas the acclimatization response is to shut down transcription. When we measure fat oxidation, and we measured fatty acid oxidation enzyme activity, again lower in the Sherpas, and this is using fatty acids in the Ouroboros oxygraph. We find it was lower in the Sherpas. And although it doesn't reach significance, there's a perhaps trend for the lowlanders to decrease fat oxidation over time at altitude. So lower fat oxidation in the Sherpas. We also found a lower mitochondrial, this is total mitochondrial capacity when we saturate with substrates, again lower in the Sherpas. And this is the leak, this is the inefficient use of oxygen that I alluded to earlier. Again, we find this is much lower in the Sherpas, disproportionately lower in fact. It does come down in the lowlanders. You are shutting down that inefficient use of oxygen, and that does improve efficiency in lowlanders, but it's notable that the Sherpa mitochondrion is more efficient even at baseline. So more efficient use of oxygen, it seems to be the order of the day. Looking at glucose metabolism, what we find in the lowlanders when we carry out oral glucose tolerance tests is that the area under the curve, so the rate represents the rate at which glucose is taken up into the tissues in the standardized test, falls. The lowlanders become glucose hungry. Fat oxidation is falling, they're taking up glucose much more rapidly and accumulating intermediates in the glycolytic pathway, so really switching towards glycolysis. The Sherpas, we don't really see this, but we do see that they have a very high capacity for the production of lactate, which may be beneficial in exercise, that they have the capacity to increase glycolysis and lactate production. My final slide is really to give you the oversight. What does this all mean, we're often asked. When we look at the energetics again, we look at that phosphocreatine energy reserve, we see that this falls in lowlanders with altitude, very dramatically falls in skeletal muscle. ATP levels also appear to be on the wane the more time you spend at altitude. So you're eating into your energetic reserves. The metabolism isn't keeping pace with ATP demand in the tissue. In the Sherpas, however, and somewhat surprisingly, because we're taking them to altitude, we're also putting them in a hypoxic environment, we find that phosphocreatine levels increase and ATP levels increase in their muscle. So despite the fact that they are becoming hypoxic, their energetic reserves are actually increasing. Finally, we're looking at oxidative stress markers here, two different markers, but hopefully what's clear is that in lowlanders, and although there was a great spread actually, some individuals see a huge increase in oxidative stress, others less so, we find that oxidative stress generally increases across the group, but falls with acclimatization. So part of the acclimatization response is to handle this free radical load. In the Sherpas, however, there was no increase in oxidative stress when they arrive. So to summarize the metabolic changes, mitochondrial capacity falls in lowlander skeletal muscle with a prolonged stay, particularly at extreme altitude, whilst cardiac energetics also fall. Sherpa skeletal muscle's got a lower fat oxidation capacity, but enhanced efficiency in terms of the mitochondrial efficiency in comparison with lowlanders, in association with a variant in PPAR alpha and lower PPAR alpha expression in the muscles. And finally, the Sherpas defend muscle energetics at altitude and show no apparent signs of oxidative stress, unlike the lowlanders. So just to finish by thanking the many colleagues and collaborators, particularly those who carried out some of the work, James and Alex at Base Camp, and my many colleagues in the Xtreme Everest team who supported our work for more than a decade now, and it's hopefully more than a decade to come as well. Thank you very much.
TopMed Talk. Don't forget you can meet the TopMed Talk team, ebpom.org/meetings. Our next big event takes place between the 28th and the 30th of September. EBPOM USA Chicago Masters Course, a perioperative care practicum.